Bonjour, bienvenue. Comment cela dit? That was just the start of my French introduction. Philipe Achambealt was here last semester, and he gave a seminar. He is visiting as a researcher at Hopkins Marine Station, where he's on sabbatical from the University of Université de Canada to Quebec at Rimouski. But he is very excited to be soon moving universities to the University of Laval in Quebec City, where he's going to be a professor, and he's looking for a few PhD students. It's good if you speak some French, but it's not mandatory, but if you be willing to first learn to curse in French, then you learn to do conversation, and then science? No, so science first.
Science first, yeah.
Okay, but he spoke last semester on his work up in the Arctic, and we asked him to come back because we have a number of you students taking an aquaculture class, and that was another seminar that he was had offered to give. So I'm going to let you take it away. And
thank you very much
for coming back.
Thank you for this invitation. Like Dana just said, I have two main topics in my lab. One was the Arctic research, which is 2/3 of Canada. And the second one is aquaculture. Since the cod fishery just decreased, you need to find something else than to look at after cod. So I'm looking about aquaculture. So having said that, I'm going really to talk about aquaculture, but not to grow species, but mainly about what we call environmental carrying capacity of bivalve aquaculture. So in your class, you've probably seen a better graph than this, but all aquaculture, that's the trend in world aquaculture production. And you see aquaculture is increasing since the 1970s to 2008. And there's also even, I found out the new one for 2014, which is even higher. And mollusk, it's one of the major growing species so far that from these report from the FAO. Bivalve are increasing around the world, everywhere. It's basically based on this. It's really simple to grow them. You don't need to add anything except some structure in the water. You don't need to find their home food. They are filter feeders, so they eat the food which is passed close by. So I'm going to talk a little bit about different type of carrying capacity in aquaculture. First of all, there's the physical, production, ecological, and the sociological one. And we realize that most, if you're in Europe, France in particular, and Spain, they're focusing a lot on production. In other words, how many bivalve you can put somewhere for the amount of food. The physical one, it's only if it's not deep enough. You cannot put, for example, mussel line. You need a certain depth. You cannot put them only in some area. So that's the physical carrying capacity. It's relatively simple to evaluate. The ecological one, that's the one I'm going to talk about today mostly. And the sociological one, for example, one of the best places in Canada to grow mussel will be a place where there's some whale watching. So no way you're going to be able to grow a mussel there because the pressure, the social pressure, it's too high for this touristic activities of whale watching, even if there will be the perfect physical, perfect production, and the perfect ecological side. But the society or the pressure, it's too high. So that's another one that we need to look at. I'm not going to talk about it at all, but that's many of the work we have done. It's also based on this one in some area in Canada and based on some law. There's a law saying that no net loss of productivity with any human activities in the marine system. So when you look at this phrase, it's quite a powerful phrase or law in Canada. So no net loss of productivity. So what does it mean? Is it and you cannot look at the species itself that you grow. So all mussel or farmers in the ocean need to compensate for the loss of productivity they are doing. That's the way it should work, or that's the way it works. So we're working with a lot of farmers to look, are they really losing productivity when they create a mussel farm or fish farm, etc.? And that's become mostly on the ecological carrying capacity. So I just mentioned these three, and just go, again, fast. The physical one, production, and like I was mentioning, the ecology. Some people believe then aquaculture is really negative. That will be difficult to create a farm somewhere because the pressure, the social pressure, is too high. So this feedback, and if you can show that it's done in the right way, you can decrease this negative feedback here to a positive one. So again, I mentioned this physical one, so that's just an example. Then it's all these characteristics. Then we can just map them physically and where we can put, where is the best area to grow. I will keep the example here about mussel because this is what the major, after salmon in Canada, it's mussel aquaculture, which is the major species that we're growing. If you look at these carrying capacity, and we're working a lot with people in France for a long time, they look only at these production carrying capacity. How many mussel, how many oyster you can put in one bay in France, then they will be on the market the fastest as they can. That's what they were looking at. So they're using optimizing the level of production. But we know it's depending on the physical carrying capacity and the ecosystem function. In their case, they use only the plankton, zooplankton, nutrient, etc. So this kind of model. I'm not going to talk about that because we're realizing with time then, especially in France, they have a lot of trouble presently to keep their mussel and oyster aquaculture. For one reason, they still increase the production, but you will see from area. So now they cannot sell most of their time, their production. So their animal rates are growing faster. They create a really nice carrying capacity with their model, etc., but they cannot sell it. That means the environmental carrying capacity is just, you're above your environmental carrying capacity because you're creating more and more red tide and you cannot sell your production, which is not better. Even if your animal grow faster, if 6 months per year, especially around Christmas, you cannot sell anything, that's not good for your industry. So we need to help them at some point to decrease their environmental carrying capacity, even if the production is really high. So this is a really simple food web. Let's say you're growing mussel in the water column here. I don't want to go into detail, but that's all really simple one, simple food web, including the mussel that you're growing itself. So for the people, and probably from your course, you've seen that, that's an example of mussel. When you want to create a farm somewhere, usually the people are using an area with a soft bottom. They had mussel socks or lees, we call that, less socks in Canada, we call that. They had they filter, we don't need to add anything in the water, and they create these biodepot close to the seafloor. I will say with a negative footprint here, and with new species, and at some distance, it's almost natural. That's the general idea behind. So basically, you create a 3D benthic system. So if you're a benthic person, you will love mussel aquaculture because you have now not only a 2D one, but you have a 3D benthic system. There was no benthic animal living there before, and you concentrate the organic matter in one site with one footprint. Based on this, probably you've seen this graph from Pearson and Rosenberg. Everyone are probably familiar with that. So Pearson and Rosenberg mentioned that if you increase the load of organic matter along this gradient here, you will change your benthic community from really rich one, decreasing the size up to only bacterial mat. I had these. So if we increase, there was no aquaculture before, really few organic matter falling to the seafloor, and suddenly if you increase your density of mussel or for your aquaculture, you're basically along this nice gradient from Pearson and Rosenberg. Can we test this in aquaculture? Is it true? But why bother? Stephen was mentioning about blue carbon earlier when we talked. I don't know if he's here. Let's see him. Yeah, sorry, I was looking this way. I had this only for you because that's true. Now, some countries are using mussel aquaculture in the blue economy, saying that mussel are capturing phytoplankton, and they are also doing some carbon sequestration with their shell. So that's part, and some countries, Sweden, for example, start to use that to compensate about carbon emission. I don't want to go into detail here, but that was to answer some of his question. There's also more and more regulation, like I mentioned in Canada. There's no net loss of productivity of human activities, but also there's more and more eco-certification, even in the area of Mont Saint-Michel in France and in Brittany. They create their own label of mussel from the Bay of Saint-Michel, which is well known, and they guarantee a color, a size, and etc., a full suite to get their, also at the same time, their eco-certification. And they sell it a few bucks higher than any other place in France, just because there's eco-label of their, I would say in French we say appellation contrôlée, like the wine. Champagne is one, Bordeaux is another one. This is what they're doing with their mussel now. And they can increase their, and decrease their impact on the ecosystem and increase their benefit because they have a label. But it's more complex than that. For example, that's Prince Edward Island, east coast of Canada. Mussel, it's more complex than fish farming because we don't know how much food is in the water column all the time, and it's changing. Can cover a really large area. For example, in one bay called Tracadie Bay, we have more than 2,000 kilometers of rope just in one bay of 5 kilometer wide total, and close to 60,000 block of cement on the seafloor to be sure in all these aquaculture. It's one bay. That's quite a lot of structure in this little bay here. And create a lot of physical structure, just like I mentioned, with 60,000 block. So we know there's some impact on the seafloor, on the sediment, and the animals. There's also impact arising from the physical structure. Are they acting, all these 60,000 blocks, are they acting as an artificial reef? There was a soft bottom before, and we have 60,000 cement block in one bay. That's quite a lot of rock, suddenly. There's some impact on the water column, and also they want to have some specific species, some specific population, so a lot of fishermen or farmers are doing some transplant, catching some spat in one place, bringing them in another bay, so bringing at the same time potentials for exotic species. So it's really more complex than we think at the beginning. So this little one, it's not that simple, because in reality you can add this now to this food web. Simple one at the beginning, and suddenly you have a full complex. I said we changed it, I will call it a Benthic 2D to a Benthic 3D system. So we have now the Benthic community associate with your mussel that you're growing, and the same thing with all the reef, all these block and ropes and etc. that you're adding in the water column. You have also a full suite of a little ecosystem that's building around it. So what we have decided over the time with my lab, it's to look at all these different compartments. So I'm going to look first and show you a few about only the bottom one. So if it's close here, what the mussel, everything doing to the seafloor. So this one, and how these little reef and bottom are changing the ecosystem. So first of all, that's the east coast of Canada, US, Canada. That's we call it the Gulf of St. Lawrence. There's an archipelago called the Madeleine Island. This is in the province of Quebec. This is where most of the aquaculture is done. So we decided to work on one site in particular, and we placed plenty of different sediment trap at different distance of the mussel. So that's a 1 year old mussel, that's a 2 years old, or we call that O+, 0+, and 1+, because it's take 2 years to send them to the market. It's quite long because the water is cold, and there's 4 to 5 months of ice shelf all the time. So it's not easy, but if you're aquaculture, a farmer, you're really happy during winter because most of them are using their truck, their pickup, and make a hole in the ice, and they can take them. It's easier and faster than to use their boat, and it's cheaper because it's their pickup. So they, from the ice straight to the where they transform the organism or the mussel to send them to the market. They love that. But there's some window it's more difficult during the high smell time. Definitely they cannot sell anything, even in their boat or their. So we, in this case, we just placed plenty of traps positioned around different direction of each of these 1 and 2 years old mussel to see where are going the feces and pseudofeces. So that's just an example here of the different direction, northeast, northwest, northeast, southeast, southwest. You have the direction of the current direction. You see that all the current are going in the southwest direction. And that's the different distance, 0, just under the mussel line. And you're at 30 meters. And as you can see, even for the southwest, where you will expect more advection of your feces and pseudofeces, already around 12 meters, it's only the background of what is in the water column. And when you look all the other one, it's really when you're under most of the different direction, the northwest, just here. So more feces and pseudofeces under at 0 distance, but as soon as you're really at 3 meters, it's a background effect of the water column. No really effect on the northeast and southeast, just this direction, quite similar at 6 meters. So really it's localized. The effect of this footprint of organic matter to the seafloor seems to be quite localized here. So at the same time, we decided to use these wildish picture. You know, it's a V shape, so you get that inside of the sediment, and you get it back on your boat, and you take a picture to look at the redox potential, which is the black line here. You can see it, and that's the different distance. And that's 2 different mussel farms, and that's 20 centimeters each one with a picture. And you can see then on this one, there's still all this part, there's a black line. But what we realized for both, at the same time, we did some benthic community. When you look at Grande Entrée, it's GE, this bay here, you see then when you look, it's quite dark black compared to this one where we have the dark line. It's a little bit lower, so this one have more oxygen in the sediment here. But when you look, that's a MDS plot. I don't know if everyone are familiar with that. If the people are not, so 2 dots which are really close to each other, that means all their benthic organisms living there are really similar in abundance and in their composition. 2 which are really far, that means they are really dissimilar. So you can see then when you're at 0, they are quite dissimilar to the rest in their benthic community. But we cannot see any change here in the redox potential because the line, it's really at around 10 centimeters, between 7 and 10 everywhere. That means the sediment is quite well oxygenated, except close to under the farms, but we don't see anything after. And on this one, you see then there's no real pattern for G, but you see then it's almost black. There's the redox potential was already, I will say, not a good indicator because everything seems to be impacted. But you've seen from the one graph before, then the feces and pseudofeces seems to reach only 6 to 9 meters, but definitely at 330 meters, there was no more impact. But if you look at this graph, that means it's quite impacted using the redox potential. So in our case, we figure out then benthic community seems to react more rapidly than using the redox potential. Why we decided to do this experiment during a master project, it's one indicator that the Canadian government or Fisheries and Ocean Canada want to use was the redox potential as the measure of impact of aquaculture. But they said, look, there's no impact, and mostly from here to here, but if you use the redox potential, you will see it's all impacted. So definitely benthic organism seems to be a better indicator if it's longer time to see, identify, etc. It's a better indicator. During the same experiment, we look at the general abundance of the species, the biomass, and the number of species. And something really was striking to us. So you see here then the abundance was exactly like the model of Pearson and Rosenberg. We have, at the distance, we can say there's less organic matter reaching the seafloor. There's less and bigger animal, but it's not at 0 distance. We have less abundance, which what we expect, and this increase quite rapidly with no change after. But it's this one here which strike us. It's why we have a small biomass at 2 different distance, especially the one at 300 meters when we're sure there's no impact. So at some point we said, okay, maybe it's an artifact. We've done that in the second farm. It was exactly the same pattern. So we said, is it possible that suggests a positive impact? And when you're really under, that's too much organic matter. When you're at just at this halo distance of the farm, it's even a positive impact. And eventually, you become to the background, which is this level. So negative and potentially positive. So that was our hypothesis, or this is what we suspect at that point. So to test this, we decided first to do a dose response project. So if we increase the stress by increasing the density of muscle or the density of organic matter, that's what we should suspect, aligned, and the responses will be an increase, an impact, a linear one, or could it be just an impact, and suddenly at certain distance, it's increasing and being positive in this case. So is it a threshold? So let's set up this experiment. So we decide to use some big trash bin. We cut the bottom and put them in the sediment like this and create some different densities of muscle in cages like this for a month and a half to see their impact and how this is going to change. I just want to remind you then that's a really big lobster. That's a new structure that will be part of my talk a little bit later, because that was one of the observation. Each of these trash bin got at least one lobster when we came back a few days after. So the density with no muscle, 200 muscle per meter square, etc., like this, up to really something that you will never see in aquaculture. To be sure, then we have the full range, and we have even a control one which have no garbage bin, nothing, but it was on the seafloor, and we repeat that. So it's 8 density, we have a control, and we have 5 replicates of these. It's a total of 45, and I said a month, it's 60 days in the water. So we look after 60 days of how much proportion of organic matter was there. We will suspect an increase. We see an increase. It was not as clear as we expect at some point, but definitely there's an increase in general, but not perfect. That's ecology, I think. And at the same time, each of these, why we use these trash bin, is because we can also use some incubation, some really incubation system to look at oxygen and different flux to see how much of the ecosystem is there's some intake of nutrient or output of these nutrient, influx or exflux. So you have the density here, and just for just one or two example, we have the PO4 here. So the density that most of the aquaculture farm are using are around 300. So really that's something that we don't make up here, but it's happening exactly where we have the reference, 0 and 200 muscle per meter square, and around 300 we seem to lagoon at least. Then you have output and intake here. So when you look at the ammonium, so same thing, it seems to have an increase in general, but it's not that clear. Then the cut is here, but still there's an increase in general, around 300, 400 muscle. We look also at these famous MB indicator from Angel Borja, and that's our reference, our different density here, the different portion of these different order. So, and you can see there's an increase of the opportunistic species compared to the sensible one, which are decreasing in general, the green here. So it's fitting their indicator. For example, you look at one sensible species, you see then it's really abundant when there's not enough of muscle impact here, and suddenly there's almost none of these Stellina agilis. And this worm Capitella, which is an opportunistic species, you can see an increase with increasing muscle density. all this you see, and there's nothing really new of what I'm showing you here. There's plenty of papers showing mussel aquaculture got an impact on the animal living under a mussel farm. So there's nothing new here. But this is where I want you to remember what I mentioned about the lobster, which is there. So we decided to say, shit, there's lobster all the time on each of these trash bins. At least one, sometimes two, sometimes four. So there's something, and this is the idea about artificial reef that came up to mind. So that's the normal system. We have these blocks, changing the current, changing also the animal living there. This is what we've seen in general. And suddenly, when you look at a block, there's plenty of other stuff, which most of the time, at least in Canada in the law, they never consider all these animals. They only consider what is living on the sediment. So this is why they said there's a loss of productivity when you start a mussel farm, because they're looking at the negative footprint on one group. So we decided to have more this holistic view, because after eating so many lobster, then we're tired and digging under our experiment, we decided to test them. Are they really more lobster on some of these mussel farms? So we went to the Prince Edward Island, the area where is the most lucrative lobster fisheries, and it goes well in Canada. It's all along this coastline here. Foresight, inside of each of these farm, we look at inside 50, 100, 500, and 2,000 meter. We did 5 transect of 50 meter distance 3 times during the year, and we look at the abundance of macro species with divers. So this is the result. It's the same trend for the abundance per species, the richness and the diversity, exactly the same trend. So I'm going to show only this one for the total abundance. So that's a different mussel farm, the 4-1. And you have the different distance, this 5 distance. And I just want to point out here, when you look directly under the mussel farm, there's always more of these macro benthic species compared to different distance. Not on this side here. So we were quite surprised, and we look. If you read the famous paper of Hulbert, 1986, I think, about pseudo-replication, he mentioned that you need to look also sometime. It's called that demoniac intrusion. I don't know if you remember that. So that was a demoniac intrusion. We couldn't do anything. The mussel farmer decided to remove all his mussel 2 weeks in advance. So, and we went there was no mussel at all. But we look, and there's no sign. So we select all these one are 2 years old, and this one was basically with nothing, only the structure. So question mark, where there's no, so it's maybe not the structure itself, it's maybe the mussel which attract the animal there. That was a demoniac intrusion, but at least we have a new question that we can answer. So what we decide to do is to look at different age of these mussel. If it's new one, where you remove all the mussel, you have only the rope and the structure, that will be what we call the M1 here. The mussel here, MH, new one, the mussel of 1 year old, 2 years old, and 3 control. So this is what we've done. So recall it's collected M1 1 year and 2 years. So when we look at the same species, then we were looking at the other graph, but we compiled them on the other one at the total abundance. In this case, we look them by species. And again, you look when you're under the 2 years old, all the time these commercial species, no, it's not a commercial species, but these 3 here, there's more abundance in general of these commercial species under the 2 years old. Not that much under the 1 year old, except for the lobster, but all the other ones. So 2 years old seems to be the time then to these animals to come to your side, or is it because there's the more mussel? So basically your food is there because they are eating a lot of mussel. Same thing, not this one, but this one is eating its favorite food, and it's a picture of an adult, but what we're looking at are really juvenile. And juvenile are really, they like capitellid polychaetes. If I finish my beer, I can do some imitation of a small one when they eat, but I'm not going to do that here. So from this graph here, we decide to look at, okay, there's more lobster. It's a major fisheries. How they are distributed under a mussel farm now. So definitely, when you look at the broad picture, there's some negative impact on the fauna, but it seems to have positive impact on some of these species, specifically some commercial one. So we decided to do this transect along in diving along these different mussel line. And when you look at the distance from a mussel line, and there's a mussel line every 10 meters, there's a new one at 20 meters, sorry. So 10, it's exactly in the middle. So when you look, that's a proportion, again, mussel of 1 year old, 2 years, and just collected. And when you're close to the structure, it seems to have a higher proportion of lobster as soon as and it's decreased quite rapidly when you go away from the structure. So definitely they are attracted by the structure itself. So really the question is it the structure or the food? So we set up an experiment, and I really like this picture when even the lobster is using the mussel line as a place to hide. It's almost close to the seafloor. There's too much of it. It's at the end of September when all the mussel there's a lot of species on it. There's a lot of biofouling. All the mussel line are really deeper in the water, almost close to the seafloor, and suddenly these animals just hide under their refrigerator, I will say. So we set up this experiment to try to understand, is it the mussel or the block itself, which could explain, or the interaction between both. We explain why there's more lobster close to mussel structure. So we did this experiment where we put some, it's a 4 treatment. Is it the mussel? So we have mussel and block here, only block, no mussel, and only mussel here. So all these 4 different combinations, 3 replicate of each. We look at that for a few weeks, 7 weeks total. So we have the different color for block and mussel in red, and the green one, not the green one, but I'm colorblind, sorry, blue. So this color here for the control, so none of these 2. We looked for 2 weeks before adding the experiment. We went on this side. There was nothing on the side. It was only a flat mud bottom, just to see, and you can see that each time we went there, all our treatment and the number of lobster are quite concentrated at the same level. 0 is when we decide to add all our different treatment, and we follow them for 5 weeks after. And as you can see, then only the mussel stay quite low and increase at the mid-level after 5 weeks. The controls stay relatively low in general. Block and mussel just increase here. And at the end, it's the block itself. So when you look at this graph, you can believe that block and mussel get some impact, but really the block seems to be more important in general for lobster, and it really matched what we have seen before. So this last summer, we decided to try to understand a little bit more about lobster. So we create, and that's relatively new result because we complete the experiment only in the beginning of October, and I don't have a lot of result, but I just want to show where we're going now with this experiment. That's the kind of block, and the animal just live under. So we decided to study the movement of the American lobster under a mussel farm and compare with different sites. So our first hypothesis is lobster use smaller area when they are under a mussel farm compared to the lobster outside. And lobsters spend more time under a mussel farm than if they are outside because there's the food at the same time. So it's a master project. What we did is the same island, but this time we decided to use offshore, exactly where there's some conflict between mussel farmer and lobster fishermen. There's always some conflict between fishermen in general, but at least in Canada, maybe not here. But definitely in this case, some area just around here, in this case around here, it's also a nice fishing ground for lobster. And the lobster fishermen believe that if they catch lobster, it's because of these mussel farms. So we decided to play the game and get two associations together and say, let's look what the lobster are doing. Are they staying here? Are they moving to your fishing ground? What are they doing? So this is why we evaluate these scientific hypotheses, but all these projects is based on conflict between two activities. So we have some reference site. We put plenty of receiver, 10 receiver, in 3 area of 1.8 kilometers square, 1, 2, and 3. One it's under the mussel farm. And we use acoustic telemetry to look at their lobster. That's an example. We have 60 tags of Vemco, a Canadian company. I'm making some ad there. I've got 10% of everything they're selling. No, I'm just kidding. But here, that's the tag, and we have to measure when they're passing by. So that's the receiver. And we put 15 lobster, then we capture here, add 15 here, 15, and 15 also in the middle. And we catch also some other 15 totally offshore and bring them inside of the aquaculture farm. So we have so that's an example. The student, she's processing it presently. So we don't see any clear pattern in general, but it seems to be more rectilinear. They are going out in general, which was surprising us. That some other lobster from here, and you see the hog seems to go this route in general, except these ones seems to go out this way. But they don't stay that much on the mussel farm. So quite surprisingly. So rectilinear movement in general. So we standardize the data and remove the first 24 hours for each lobster. And this is what it looks like now. And you can clearly see then, when you show that to lobster fishermen, they realize that now their lobster go back to their ground most of the time. All these ones, you can see they are going back to the fishing ground, which is just around here. These ones seems to do a little bit differently. But in general, as soon, I don't show it enough now, but many of them, as soon as they get here, it seems that they start to go back in this area because each lobster and the tag got a number, so we know which one. And each time we know that they are out, we lose the signal. But if they go by just here, we catch again the signal. And at the beginning, we thought it was some noise, so we removed that. But we decided to look at these little details, and all these seems to go around like this and come back in this area. Anyway, most of them, really surprisingly. We were suspecting they were going to stay there based on what we observed. Of these 32% that return, 10 lobster display only rectilinear movement and 9 display homing behavior. So that's not that many who stay in place. So just to test this, the second hypothesis, we look at lobsters spend more time under mussel farm. This is what we expect. And here we are, the number of hours they spend under. There's no significant difference, but surprisingly, they are more outside. They don't stay inside. And speed, we will suspect then they will be slower under the mussel farm, which is not the case. Going in more detail, she never received this question. I made this question earlier today, not today, but during in the last month. Could other factor explain lobster movement? Because definitely they seems not to stay there. And so maybe the size of the lobster, lobster are cryptic and they try to keep their hole. So maybe their goal was to have as much lobster of the same size as possible. So we don't have a big range of lobster, so it's 70 for the cephalothorax size to 90. It's not really a large range, but we will suspect that large lobster get under, and you remember these trash bin, how big was the lobster? So the bigger lobster probably get the hole, and all the other one search for a hole, but if there's a big lobster, they don't try to find, they don't want to lose energy to try to get to this hole. You just keep going until they find an empty hole. This is what we suspect, or this is what I suspect. Don't mention Emily, then I suspect that. She needs to find this hypothesis. But definitely it seems to have a small, even if the number are really small, my hypothesis, and this is what we're going to test this summer, we're going to try to find some lobster which are going to be way bigger and even some smaller to test if the size of the lobster could explain why they are seen at this range and not staying there. So I've been only I said, is that not simple? So you see, I can talk forever, but I just talk about this area so far. So now we can talk about the mussel sock, this 3D benthic habitat, which is complex by itself. So this is what looked like a mussel line in the Madeleine Island in Canada. You don't see a lot of mussel on it. This is even what it looks like when they have some invasive species. You don't of biofouling all around. So can we, in this case, we want to go back on the hypothesis then, or the law in Canada saying there's no net loss of productivity. Do we believe it's really about the mussel itself or all the biofouling? What is happening? What is the impact of this biofouling in general? So because the experiment is about no net loss of productivity, we decided to test this hypothesis at the Madeleine Island, and not in the Madeleine, and the Prince Edward Island in 8 different bays here. We have the mussel leaves, and we create our home mussel sock and leave them for the full summer. We have 5 in each of these bays, and we have control area. So this is what it looks like. We create our home mussel leaves, and we decide to sample an area here and under, and definitely nothing in the water column and only at the bottom. 8 sites, inside and outside. We look at sediment core. We look at line under the mussel, of mussel line, 50 centimeter, and we look at productivity, diversity, all these usual parameter or variables. So these are all the different mussel farm. This is the control where there's no mussel, and this is where there's some mussel. And we're looking now really at the productivity. We take word by word the law in Canada, which is the productivity, the number of kilojoule per meter square per year. So when you look now, and you see all the control seems to be higher in general than for the sediment than all the mussel one. So definitely, if you look only at what is under the mussel farm, every farmer needs to compensate for the loss of the productivity. That's what the law is saying. But when you look now at all the biofouling, at the same time, it's totally the opposite trend. So doing mussel aquaculture seems to increase the productivity of the area. And if a mussel farmer wants to use our work, now it's the opposite. They are creating some productivity. In general, you can see, as soon as we consider now the mussel, the 3D now mussel habitat, except a little bit here in this case, which it's an increase, but not that major. I'm going fast. We did some other work there, but the last bit I want to talk is about the nutrient now. So we look at the reef, we look at the mussel sock, we look at what's happening on the benthos here, but what's happening in the water column. I'm a benthic ecologist. I'm not a plankton ecologist, but we look at least what is the nutrient, what is the ecosystem functioning of inside of the water column. We create some benthic chamber. If you put some mussel line inside, we can see how much exchange of oxygen captured, and we've done that also on the seafloor. That's the kind of experiment we're doing with the mussel socks just inside here. So here we have your different fluxes, phosphate, ammonium, oxygen, mussel of 1 year old, 2 years old. It's only with your control, and here you have this line, it's only the mussel, and the dashed one, it's including the epifauna or the biofouling on it. The goal is to see what is the impact of the biofouling itself and the mussel. So for the 1 year old, you see there's no real difference except between August and September. There's a decrease for the ammonium. But when you look at for the 2 years old, and especially in September, the biofouling is increasing. We don't have a nice weather like this all year round. We have a lot of ice, I told you, 4 to 5 months per year. So necessarily, your mussel or all the biofouling will be higher at the end of the summer, which is in September, from the biofouling and the mussel itself, none really in August. So only in 1 month you have an increase, drastic increase of your biofouling. We have the same pattern here. We have a decrease in ammonium and eventually no clear pattern, a little bit, but no significant difference. So really the ammonium is higher. It's coming from the mussel itself. You see there's no difference with the inside, including the biofouling. And the same thing for the phosphate. It's higher from the mussel and not at all from the epifauna or the biofouling, which is not the case for the oxygen, which is really higher here. So I can go into detail. I'm not going to go into detail, sorry, but each time all the difference seems to be at the 2 years old when you have the biofouling. But all this difference cannot be explained by the low biomass, because we measure the biomass of these species on these, and it's not really high, but we look also at the biomass of the sediment side of these mussel socks, and this biomass was really high. So, and we know that all these fluxes are not created by larger animal itself, but they are by bacterial degradation, most of them. So really, it's probably the sediment trap between mussel line, which is really important in these change of these different nutrient ratio. Why I mentioned nutrient ratio, it's now we're working with the people in France because we look at their red field ratio. So how this red field ratio changed because of the density of mussel or oyster, and we see then it's really a direct link with the type of phytoplankton species you will observe in the water column, because we know then a lot of species are related to this red field ratio. And when we look at what's happening in France, their red field ratio, it's directly favorizing or increasing the potential for dinoflagellate, and it's linked directly to their increase in red tide. So definitely we have a link there. We're going to explore this from this study with the in France, the Lagon of Thau in France presently. So the next step now, that's another place in France where they have all these structure. It's in the Baie du Mont Saint-Michel, a really beautiful archipelagos. It's called Chausey, between UK and France. And they have these almost 1 kilometer of these, they call that bouchot, which is only a wooded sticks at high tide. They got close to 15-meter tide there. So at low tide, they go with their truck, with their stuff. And this is at low tide, and this is at high tide. So their mussel, they wrap the mussel around in 2 lines like this, 20 meter and another 2 line, and it's 4 kilometer like this. So it's like a little forest. So what we want to look now with them, almost go to what I'm just showed you before, all these different experiments, and try to test that in an interdictal system for France, and look also at all the dynamic of these potential dinoflagellate, based on these sediment trap inside of the mussel socks, or in this case, bouchot, they call it. So just to summarize, there's a change in general along these Pearson and Rosenberg. We've seen there's a difference. We've seen there's potential some increase at some point in productivity. We showed that on the last few slides. There's an increase in abundance and diversity of macrofauna. I specify macrofauna because all the infauna, we've seen it was mostly negative, and that's what the people are looking in general. For lobster, it's really due to the addition of physical structure. There's more and more lobster, and we have a better idea of the dose response. We need to clarify some of these questions, yet which level will be perfect for these red field ratio, especially for the work we're doing with France now. Yeah, I'm not good with animation, I always forget. And just to complete, I want to do a little bit of advertising. I'm the president of the next 4th World Conference of Marine Biodiversity, which is going to be held in Montreal. It seems far, 2018, but bloody I'm late to organize this. That's the website, just here. If you want to follow us, we're going to, in the next 2 months, that's going to be faster and faster, all the information on these Congress. If you've never been to Canada, it's maybe the time because someone want to build a wall between the two countries. And that would be the right timing, 2018. That's in Montreal. Montreal, it's a beautiful city in May. So don't hesitate at least to have a look sometime. Thank you very much. Question, comments?
How did your findings on the lobsters associated with the reef-filled fishermen, and has the conflict shifted, or?
It shifted, definitely. When you work with lobster fishermen, it's the most lucrative, I would say, fisheries in the east part of Canada. So lobster is really a sensible species. When I'm saying that, we see a decrease in general of lobster catch. So we know that it's probably a lot of overfishing in general. They are fishing by number of cage, not by number of landing. So each fisherman, for example, owns 500 cage. So it's a day. It's not if they catch everything the first day, they still have this number of days. So it's not by landing, which is sad. So that's difficult to regulate. So if they fish and fish and fish, that's the number of days for a number of cage. So they believe that's now the law. What we have now, what we're trying to deal with is that no, it's nothing to do with the habitat. It's the mussel which are eating the post larvae of lobster. So it's another reason. So now we're going to test that. We have already tested for us. Now we need to test it with them. We're going to bring the camera. We took some of their lobster. We took some of the mussel for the sandbait. We're going to do some really, I've got a footage. It's so obvious that it's the mussel which is stressed, not the lobster post larvae. You can see the post larvae, which is a little predator, and goes and starts to eat the mantle of the mussel. So that's what we're going to show them after the lobster season this year. But they need to be physically with you when you're doing that because they're not going to trust you. There's a lot of, in Canada, following the cod fisheries, etc., the collapse, there's a lot of, they don't trust each other. It's difficult. This relationship is not easy. We need to rebuild it. And this is what we try to do, bring them, tell us what you want us to do, and we're going to help you to get there on both sides, to be sure it's a trust relationship to gain that again. So that's why they shift that now. It's the mussel, it's still the mussel, because mussel aquaculture is doing well and better. So they can see it. I'm catching lobster, they're selling more mussel. Shit, they don't like that. That's the way it's probably human are doing. So it's their responsibility, it cannot be mine. So now we try to show that then. Can you decrease the number of cage? That's what we want to know from them. If they can decrease the number of cage, you can still catch them, but yes.
In your lobster experiment where you look at where they move with the lines that you were investigating, were they 2-year-old mussel lines?
Yes.
Okay.
Yeah, we use only 2 years old. We've seen from the beginning that 2 years old seems to be they're really the one which makes the stronger difference. Yeah.
And then when you leave that territory to go to where like the lobster fishermen fish, is the benthos there more rugose, like better for lobsters? Or?
That's our big question. That's an excellent question. What we suggest so far, it's lobster, they don't really like mud flat. They like when it's pebbled, when there's some rock, etc., some kelp, laminaria in our case. So in the area where we are, there's very few because it's a mud flat. So we believe that they are trying to go back to their original perfect habitat, which is where there's some kelp, etc. So that's what we believe. Probably go, that's the closest distance from the mussel farm. It's only just north. So that would be what we suggest. But it's a question mark. Is it really only that? We don't know.
Did you see any potential evidence for another species of animal that lobsters might not like that does also hang out under the mussel cages that might be deterring the lobster from going under there?
That's an excellent question. Never thought of that. I know lobster don't have a lot of predator in the East Coast, so I will it's not a predator. And they are the dominant predator of crab. We have the common crab, so they are the dominant. I'm just making that. Yeah, I think that's an excellent question to explore. Which group can be different? But definitely there's very few kelp. That would be, maybe it's missing something. Not only another species, but that would be a really good point to explore, because where there's some mussel, there's very few laminaria in our case, or kelp. So maybe it's a lack of one species that can be. That's an excellent point. Thanks. Some other question?
Question? It's happy hour.